All right. So welcome, Forrest, to the Coronavirus Multi-Species Reading Group. It's a real delight to engage with all these different texts. On the table for discussion today, we have your lovely book that is a guide to phage, life in our phage world, a centennial field guide to Earth's most diverse inhabitants. We've also got a paper that you're the senior author on called Viruses as Winners in the Game of Life. And I've also been reading more broadly, you've got amazing papers also on the human virome. I know that I've heard you on TWIV recently talking about the sequencing that you've done of the human home. And one of the things that I thought I'd start off with is just in the field guide book, you talk about the language of ethology as a way to approach viruses, which struck me as really interesting and original. And I was wondering if you could say just more broadly about what it's like to think about the dynamic nature of phage as creatures that have behavior, and maybe by way of background into this question, early on in the pandemic, we read a paper by the philosopher of biology, John Dupre, who basically, and his colleague Stefan Goodinger, who insists that we kind of abandon the distinction between life and non-life as we approach viruses and think about viruses as living processes that interface with other processes on multiple scales. So as you approach phage and I guess viruses more generally with the tools of ethology, what kinds of interesting things are emerging?
Yeah, so I might turn it around to you guys at some point. So the interesting thing, as you probably know, with phage and viruses in general, people tend to really think of them mostly just as parasites or predators, right? I mean, not really much about all the other cool things that they're doing. And then, of course, the molecular point of view would just be how they affect metabolism and so forth. But of course, they're really, there's a lot of them out there. They're astronomically successful. So that means that they're doing all of these things. And the reason that it's sometimes useful to think about what their behavior is it gives you a different way of even looking for what they might be doing. Does that make sense? Like literally just applying words to things that we might be able to study in the lab or in nature. So that's actually why. And in fact, we've tried multiple times and you guys may, definitely the philosophers in particular might have a better way of approaching it, but really just thinking about like different things that we would see animals do or humans do. Could we actually do the viruses also do that? And then are there things that we can't even imagine animals and humans doing that the viruses are doing that we could be then going and looking for? So that's really the idea. It's just to open up the places that you look. You know, for the most part, basically, people only say viruses for how do they kill a cell, right? How do they get around? That's about it, right? I mean, that's their main thing. They're doing all these things. And we know this because we know that about 80 to 90% of the genes that they're carrying, we have no idea what's going on, which means that we haven't looked at them correctly, which is, I think, a lack of imagination on our part as scientists more than anything else. Do you guys have any great ideas for places, things you would look for, right? Does that make sense?
Totally makes sense. And I mean, there's definitely some conceptual resources in our field, broadly construed. So there's a classic paper by Karen Barad now about brittle stars, stars which don't have an eye or don't have a brain, but they are eyes. Like their exoskeleton is like, you know, a visual array. And Barad uses the language of well, she insists that we get beyond this idea that, you know, there has to be a distinction between knowing and being. That, you know, when you look at these organisms that are organized according to, like, you know, these nanoparticles that do the work of response rather than, you know, centrally coordinating, you know, the input comes in, it goes to a brain, and then the output goes. to think about ways that, in her terms, interactions take place, where these dynamic exchanges happen at that molecular level. So, as the paper by Mary, am I saying her name right?
Yeah.
Mary Ewell?
Yeah. It's worse. It was Mary Carol Ewell.
Mary Carol Ewell. And I must say that, I discovered her work, and it's an experience I haven't had that much before, where you discover someone and then realize, oh, they just passed away. So it was such an amazing encounter with someone who's recently deceased. But her paper, with this T4 phage dancing across the surface of E. coli just really made me think of Barad, and really made me think of the liveliness. So that, and she's got a very profound question in there. Are viruses sentient beings? So the concluding line of that essay, in actuality, they are a sophisticated and economical mechanism enabling inert virions to respond adaptively to diverse external cues. They raise the question, are T4 and its relatives sentient beings? Which is a really interesting, destabilizing question that I think most people would be totally unaccustomed to. So how does Mary approach that question, and how do you think about these kinds of things?
Yeah, I mean, it's it would really get down to I'm going to hedge a little, right? What we want to call sentient. What I do think is that they're much more alive than we normally give them credit for, and I mean that in the colloquial sense, right? They're doing lots of behaviors that we would be really amazed if we could observe them, right? I mean, they seem to be, global travelers. They seem to be, really changing their environment around them to make it better for themselves. They definitely have, literally planet-wide sort of influences, which is kind of cool, right? I mean, really, you don't see a lot of organisms doing that. I mean, noticeably people and bacteria is kind of a group, and viruses also is a group. I mean, that's always what's unfair about viruses, is we tend to all put them together whenever we want to make them sound really cool, right? But there's of course some that are probably not very important at those scales, and then others that are very important at that scale. Yeah, so sentient, I'm not sure, but something past what we would call, they're not just inert particles either. They're definitely reacting to their environment and reshaping their environment to make it react to them.
Super interesting. And promiscuous, that's another word that you use. They're promiscuous and engage in kinky sex games. For example, homologous and illegitimate recombination with related and completely alien genomes, orgies of hundreds of genomes. And also, not only mixing genes amongst themselves, but transferring amongst other organisms. You talk about the holobiont and phage and viruses as kind of the key bionts that animate these relations amongst holobionts. Do you want to say something additional about the promiscuity here?
Yeah, so it's kind of, it may be like one of the, like the normal evolution we think of, like descent by modification sort of stuff is not, it may be that that's like just a very localized phenomena, and that really what's going on is you're getting recombination of different genomes, and particularly viral genomes, constantly, and those are creating, in the lexicon of like, you could say hopeful monsters, right? Things that come together in some really weird combinations are happening probably all the time. And that's because whenever a virus goes into a cell, it's going to run into another virus. And when that happens, really, honestly, from the DNA point of view, chaos ensues, right? So they start cutting up the host DNA. They start making their DNA. So they have polymerases and they have, exonucleases and endonucleases all doing all of this stuff. And that leaves a lot of free ends of DNA to run into each other and then be extended and put, so you're mixing and matching all the time. And the fact that that's happening really at 10 to the 27th, which is like our guesstimation of how often this happens per day, you're working at such large numbers that you could have the kind of I think there probably is just like this global virus sphere that's exchanging information all over the planet, and it's doing it all over the time. And so that's a very different way of thinking about evolution. Because one, you're putting things together that don't normally go together. And two, you actually could have selection at very small levels, something that we could never measure in the lab. So if you, like roughly speaking, let's assume that the number of viruses in a population, for whatever that means for a virus, but let's say a gene out there in the virus sphere is about 10 to the 23. So that's basically Avogadro's number of that gene on the planet. And we do think that that's about the numbers that they're happening, which means you could have selection pressures that are incredibly small and you would never observe them. There's no way to, in the lab, to do that, right? So that gets you into this completely new way of thinking about how selection would work and how you could have things jumping from literally from the ocean to a person, right? There's no, in fact, there's kind of evidence that that's already happened, or there is evidence that that happens, and it's probably happening all the time.
Super interesting.
Yeah.
And for the folks that don't have the background in molecular biology, if you could slow down with those molecular processes. So you talked about proteases, endonucleases, and exonucleases. So what are those in sort of for someone who hasn't taken a cell biology class?
Yeah. So imagine you got a cell and you got a virus, and it comes along, and first they kind of taste the outside to make sure, is that a good cell that they might eat? And then once they think it is, they do kind of that specific attack, and then they inject their DNA into the cell. And now you have the virus genome inside the cells with the cell's genome. And that cell almost always will also have another virus, right? Which would be a provirus living in there. Now, what happens is that the invading virus tries to take over the host cell and turn it into a viral factory. But the virus that's already there is like, oh, I'm going to die, right? So it starts to go into its active phase, right? So it starts to activate. And both of them start to chew up the host DNA because they need the nucleotides, right? So they need a whole bunch of, the nucleotides are what make up the DNA. So they need that. So they start chewing that up, which means that suddenly the DNA that was one big long strand is now hundreds, if not thousands of strands. And that allows you to put then when you start stitching the DNA back together, either to copy it or where you just get a piece of DNA sticking slowly to another one, there will be a polymerase, which is a DNA polymerase, which comes along and it makes the next copy. And if that happens where weird things got together, they'll actually be stitched together, which is completely, it would be analogous to taking a book, like many books, and cutting them up and just randomly pasting them together, completely the same thing. And that's happening all the time, right? Like as a constant phenomena on the planet. So that's really a very weird thing for biology to work against.
Very interesting. And maybe just thinking with the holobiont for a second.
Yeah.
So we have eyes, we have like this haptic awareness of like we can touch other creatures, but they have fuzzy boundaries, and part of what you're describing here is the constant traffic that destabilizes these things that we think of as stable, which is quite a radical thing. In part, I'm maybe curious about the endogenous viral elements in the human genome, and we know it's there. We know it's probably changing, I would imagine, as these new viral elements interact with us in real time. What can you tell us about getting sort of going from we got the molecular and we've got the macro. If we focus on the holobiont for a second, talk about that as a concept as it wiggles, with these top-down and bottom-up dynamics.
Yeah, so that's kind of one of the, or that's a little of my bread and butter. So I really love that idea, right? So your intergenerational adaptations or acclimatizations, people may use that term, is almost certainly driven by the viruses, right? So what is happening is you and I will live, let's say, 70 years, but over the course of that time frame, we'll see different conditions, and you need something that's adaptable, which the human genome would not be. So what you need is something that can come in and rapidly respond to changing conditions. And that seems to be mostly driven by the viruses, followed by, of course, the bacteria. So by just changing associations, you get that to happen. And then within the human, so I don't know, you guys probably have never seen this stuff, but like so in your gut, in humans, it's easy because it's basically just our colon. So most of our microbiome is in the large intestine. And there we see the phage in particular that are produced by the bacteria in the fecal matter are actually sticking to the mucus of the human host, and they're specifically adapted to do that. And they do that within each human individually. So they have a hypermutability mechanism that allows them to do that. And so then everybody has like this microbiome, but then everybody has a viriome that's like sticking on the mucus. And that's protecting you against bacteria that are trying to get in towards your cells. Even more cool, I think, is that those viruses, our cells will pick up one of those viruses and they'll transcytose them, which means they take them from the apical side, so the part that's sticking out into the colon, and transcytose them into to the basal side, which is really cool. I'll just tell you some immunology, so you've got it. So imagine you have two tissue layers, right? So this would be the stool right here. Then there's a mucus sample, there's mucus, then there's a cell layer. And then there's another cell layer, and in between it is where your lymph is. So your lymph is what is one of the main parts of your immune system. So imagine that what's happened in biology is that at least in mammals, because this is where we know mostly, phage in particular are being taken in and they're being put in the lymph. And that means that they can go throughout the body and they can kill bacteria within the body. There's also a whole bunch of cool other things that they could be doing. So when they're going across those cells, they could, of course, be delivering DNA. We don't know, right? We know that they're cleared very fast. So they're going through cells all the time. And just to give you, again, the thing with viruses, the one thing that you should always put in your brain is just the numbers, right? There are as many phage going into your blood every day as you have white blood cells, even though people don't talk about them that much. And then, of course, when we look in the blood of a human, you have the phage, which are coming in mostly from your gut, but also from your mouth and a couple other places. But then you also have all the endogenous viruses that you were talking about. And so we have a significant number of viruses running around in our blood all the time. And it doesn't matter if you look like you're viremic or not, right? So that's unusual for us to actually be sick from a virus. Most of the time we have viruses in our blood, but they're not doing anything, or we can detect them there. And these are viruses from all over the place, including, like the nasties, right? So different pox-like viruses and papilloma viruses, and those guys are in your blood all the time, and not causing you any trouble that's apparent. And then finally, I would just mention the most underrated viruses of all, which are called TTV viruses. And they have different things that people have called them, but transmission transduced viruses transfusion transduced viruses is how people originally called them. But basically, these are just little viruses that live in everybody. And we don't really think that they seem to be true symbionts, right? They're just hanging out, or true neutral symbionts. It doesn't seem to hurt you, and we can't find anything that they help you with. They're just hanging out with you. So lots and lots of viruses.
Super interesting. So one concept that a colleague of mine, Heather Paxson, an anthropologist, developed to understand the microbiome, the bacteria, is what she calls a postpastorian microbiopolitex. So basically what that means is that, in terms of human practices, what we're looking at now for raw milk cheese producers and others is not kind of the FDA approach of sterilize everything, one size fits all, to killing microbial life, but the selective cultivation of good microbes, but not treating goodness as any kind of essential quality to the microbe itself, but to treat that as something that's situated within a particular microbial community or a particular biocultural community. And I'm wondering, like, as you're talking about phage getting selectively incorporated into the body and into the lymph, how does the organism, but before we start to think about cultural practices that are elaborated on top of this, but how does the organism make those judgments, or maybe it's an antagonism, maybe there's, if we're thinking about agency in these relational terms, maybe the virus has some intention, and the organism has other intentions, like the host. So how are these dynamics worked out? Like, is there any kind of differentiation? Is the body saying, I need this kind of phage now because this bacteria is cropping up over here, or is it chaotic, or maybe we don't know yet?
Yeah, so it is, but it won't be that terrible, right? So one of the things that's, so this is way too much biology, so you guys can shut me down or just ask questions. Please don't be shy, because it's very complicated. So as a virus goes into the host body, into a mammalian body, there's basically two ways that this can happen. It can go in such a way that nothing that's called a danger signal happens, all right? And a danger signal is like LPS, like the cell walls of bacteria, or dead human cells. That's one of the main ones, is if you kill off a cell or you stress out a cell, they start putting out these signals, and those signals are meant to get the immune system going, right? So that's like when you do a vaccine, you put an adjuvant in it, and the adjuvant is to cause the immune system to be ramped up, right? But if you put the same thing in there without the adjuvant, right, the body, instead of like reacting to it as if it's a foreign entity, it reacts to it as if it's part of the body, right? So you get tolerization. So this is actually really the self versus non-self version of how this is happening. So what we think is going on, at least in that particular case, is that you've got viruses making it into the mammal's body without setting off danger signals. So to answer your question, at least at that part, we would say that it's being recognized as self, not as non-self. And we know that that's kind of true, because if you take phage from someone and you look for antibodies to those phage in their blood, they don't have them, even though we can find the phage in there. But if you inject someone with the phage for phage therapy, they will develop antibodies to the phage. So it really does matter the route, and the route has actually changed how the interaction works. The hard part, and the thing you guys should like grill me on, is that we have no reason. So it's easy to see how the mammal is gaining from that, right? But it's very hard to see how the phage are gaining from that, right? And we would always assume that the co-evolutionary stuff should, both things should win at some point, right? I mean, that you would guess. But I don't know what that is. I have no idea, actually.
But doesn't the phage just get a host that is still alive? I mean, the bad virus kills the host and doesn't have to live. Then you have to get really big. You have to really, the math becomes really hard, right? Because it has to be some population to community level. One of these. I mean, it would work. Like you could find the math that would say it would work. Whether that's what's happening or not is a little tricky. Interesting. Well, I've got a million other questions. Other folks should feel free to jump in, either type a question in the chat, unmute yourself, raise your hand. Go ahead, Amina.
To just catch me up on biology questions. Do we have any suspicions? So if the virome, is the late surprise, or phages are the late surprise to the microbiome conversation, microbiome surprises humans a couple of 100 years ago, at least. Do you have a sense that there's another surprise beyond the phage, even less visible, less knowable set of processes in this mini cosmos, reverse cosmos, is one question. And a second question about bats.
Well, so yeah, I mean, there will be another surprise there. I mean, as you probably know, for a long time, really until I'd say the 1990s, people hadn't really thought of the microbiome too much in the context of human health, other than it being kind of a pain in the butt, right? So that anybody that was thinking about the microbiome as being healthy was really, or helpful, would be ruminant biologists, right? So they were all thinking about that part, but human stuff, definitely not until I mean, really, where people were thinking that they could manipulate it in a useful sort of way. Phage are definitely, it's kind of weird that you say that, because of course, phage are in many cases what we figure out first, but then people, everybody's so self-centric that they don't really think about the viruses, even when they, if you give a talk, I'll tell you. So if you go to a microbiome meeting and you give a talk, you'll give a talk about, you'll be the second speaker, and then everybody will go, oh, that's so cool, right? Oh, they're all excited about the phage. And then the next talk person will start with a slide about how, oh, the bacteria are the most abundant thing in the human but every time, I'm not joking. And so people just have this tunnel vision when it comes to the viruses. That's something to keep yourself, or to remind when you're thinking about, because actually we know a lot about how viruses and cells interact, even within the context of the human microbiome. The big ones, of course, are probably going to be these weird protist-like things. Every time we start looking at weird eukaryotes, they're just popping up everywhere, and we don't study them within the context of the human really at all. We don't have methods so with viruses, it's reasonably easy because you can isolate the viruses away from everything else just based on their size and their density. You can, follow the bacteria using marker genes. Protists are really tricky. You can, but not as easily. So I think that we'll find a lot more interesting human, basically unicellular eukaryotic interactions. That would be my guess.
Thanks, Boris. And
Yeah.
I do think that there are religious reasons why we see resistance to an imagination for the microbiome all the way up into the 90s and probably even still in certain communities, unfortunately. That's really fascinating. In a completely different direction, you mentioned the endogenous, thank you, I'm plugging in my computer, which is losing its power. Still I'm missing, that's not it, thanks. Sorry, I heard you say that, speak about all of these endogenous, some of them are symbiotic, some of them are not. Viruses in the human, we heard a few weeks ago about bats being far more tolerant of infection, and infection not necessarily translating to disease in a way that it's not the case for really any other mammal on the planet. Do you push against that idea?
I probably would push against that idea. I think actually we're always, like, I think all animals are basically interacting with viruses continuously. I think the bad thing is that one, everybody's tunnel vision towards bats anymore, right? And the temperature thing is real. Birds and bats really do lead to more things in humans because of their higher temperatures. And as far as, let me put it this way. When we look in a coral, which are so like if you were to say the tree of life and you were talking about metazoans, so animals, right? Humans and corals are about as far apart as you can be, right? They're essentially some people argue that sponges are animals. They're not really, they're kind of glorified microbial mats, you might say. But corals are definitely an animal. They have two tissue layers. They do all of these cool things. They behave like animals, and they have a massive viral infections. Like, they're filled with viruses all the time, and they're viruses that we find in humans. So even though these things are separated by like maybe 500 to 600 million years of evolution, you've got basically herpes viruses that we recognize as herpes viruses in corals and in humans, right? And we've even got like really conserved, like literally the responses to them. So within the animal's genome, you have how they respond to viruses are actually encoded by genes that are complete homologs. And they don't look similar at all, but if you make the proteins and you throw one, like if you throw a coral one on a human cell, it reacts. And if you throw it's, so you know what I mean? This is massive conservation. Probably we've been interacting with viruses the whole time. And I would also like even go further and argue that most of these interactions are benign or beneficial because it's been going on for so long. So you tend to get attenuation, and then mutualism seems to be the rule. And really what we study are kind of the spillover events. So either where a virus jumped to a new host, and or we created a hopeful monster that causes problems. So that would be kind of, if I was just to sum it all up. Yeah. And none of that is backed up by data, so you don't have to. We've moved into your realm of faith, okay? Yeah. So it is actually very cool that I think the viruses are really, honestly, they really are so central to biology that it's impossible to consider biology without them.
Yeah, thank you for that. Yeah, super big picture. I mean, everyone's like, who done it? The pangolin, the bat, So now we'll say the corals did it. The corals did this. Roberta, do you want to jump in?
Yeah, I wanted to. So I really love that little snippet of the book that we had to read, and I was very fascinated by the sheer number of these entities, but also I was looking at the geography and the fact that there's a lot of concentration of research in certain parts of the world and not in others. And I'm wondering if this has to do with the scholarship. And then I also have another question that has to do with like my own perception of viruses. I've been studying viruses from a cultural studies perspective for a long time, 10 years. And I always bump against this polarization of ideas, the fact that like if you look into popular culture, there's definitely like a lot of negativity about them. But if you talk with certain scholars, not all of them, you have ideas like yours, and I'm wondering how diffuse is, how spread is this? If is science changing the way in which they are seeing viruses? So like the idea of like the constraints, like the idea of the virion that is always portrayed as like this like block. Like is it changing, or is it something like is, because I'm getting a little bit confused, because I'm talking with a lot of scientists who are on my side, but then what about the other side?
Yeah, so no, I would say it moves at the speed of science, honestly, that that it is very hard to break entrenched ideas. So it doesn't really matter how, I mean, people are getting more interested in viruses, no doubt about it. And in the sense of them not just being a pathogen. However, you know, COVID and HIV and influenza doesn't help our thing, and frankly, also the phage. You know, most of the things we know about bacterial pathogens are actually caused by phage that are in the bacteria pathogens. So again, it's not surprising that there's a focus on them as disease-causing organisms. There is very little research pushing towards what are the normal relationships between viruses and even really bacteria and animals and plants. That's a little, you know, it's very hard. It goes back to what we were talking way at the beginning. We have to come up with new terminology for new things to be looking for that would be really interesting to people about possible either neutral or beneficial relationships. And to answer your first thing that you pointed out is that there's really two reasons that we mostly work in the in aquatic environments, in particular the ocean. And that's because that's where we started. So you can filter a whole bunch of water and you can get a whole bunch of viruses and you can sequence them. So that's step one. Step two, there was actually funding. So the Gordon and Betty Moore Foundation put like literally tens, probably hundreds, probably $100 million into studying marine microbes through the beginning in the early 2000s. And so basically that's why you see the marine environment being the most studied. So it's easier to do. It's actually kind of tricky to work with swales and sediments. You know, you can, but you have to adjust all of your techniques to do that. Yeah. So right now it's basically humans, human associated, and the marine environment for any real depth of it. Yeah.
Thank you.
Yeah.
So on the marine question, you know, this paper, Viruses as Winners in the Game of Life, and other publications go into the way that microbes and viruses have a tremendous importance in the global biogeochemical process. Mm-hmm. So in the same way that we were thinking about, you know, beneficial phage inside of an organism, you know, right now we might think of phage as maybe enabling conditions of life on Earth in the sense of like, you know, if we saw different dynamics of marine microbes, like whatever fossil fuels humans are introducing into the atmosphere, like it could spiral out of control. So, you know, we've got Lovelock and others theorizing, you know, Gaia. But maybe my question is, does the math work out? So you know, if we're thinking about the ways that these biogeochemical processes are being regulated, you know, how does it all hang together, and why? You know, why do these systems remain stable along long periods of geological and paleontological time?
Yeah. So that's a great question. So I'm going to do some ecology, and stop me, please. Okay, so like there's kind of this fundamental relationship in the natural world, which is that the energy sources that everything uses come from ionizing radiation. So you have to you get sunlight, or you get radiolysis in the crust, which splits water, right? And that's what gives you oxygen and effectively sugar. Let's call it sugar for all intents and purposes. And that those have two fundamental characteristics, right, that are different. So the sugar is a solid, the oxygen is a gas, okay? Which means that they decouple from each other. Okay? Now, on the planet that we inhabit as big macroorganisms and on the surface, we are dependent on a lot of extra oxygen being there, right? And that was because there was a decoupling where the sugar got, you know, squished and, you know, lost as oil or in sediments and this. And then now we have something like 18%, about 20% oxygen in the atmosphere. And so humans and human things that we like have to have a lot of oxygen compared to the amount of sugar. Microbes, on the other hand, a lot of them like very low oxygen and lots of sugar, right? So think beer or wine, right? Those are microbial dominated systems. There's no oxygen, but there's a lot of sugar. And so, and you put a human in wine and they may be happy, but they'd be dead really quickly, right? You know what I mean? That sort of thing. So, so what happens with the viruses is that, so right now, what humans are doing is we're trophically changing the system from an oxygenated system to a sugar-dominated system. So we're literally shifting everything. And we do that by like a whole bunch of mechanisms that you guys have all heard of. But the easiest is to just think of like sewage being dropped out into the thing. The most important one is actually the overfishing and the hunting, right? So as we've removed all the big organisms, we're left with small organisms getting all of the energy. Okay, so that's a long way of just saying that there's a trophic, we call that trophic downgrading of the planet. Okay? And trophic downgrading leads to a different type of viral behavior. So viruses that are in high sugar, low oxygen, tend to behave temperately. So they're hanging out as proviruses, and they tend to, so they hang out as proviruses, and then they tend to evolutionarily have a different game going. They have to protect their host cell from other viruses and from other things that might eat them. Okay? Whereas if you're in an oxygen-dependent environment or high oxygen environment, viruses tend to behave lytically, so or virulent. So they're mostly producing they're doing little bursts of activity. And so what's happened is, like in the true sense of like a Gaian sort of switch, is that we're going from kind of lytic behavior to more temperate behavior. And that's not a good deal for animals, because temperate things, that switch is what the viruses aren't killing the bacteria anymore. They're not like causing things to turn over, the recycling and everything. When they're protecting them, they're producing things like virulence factors. You guys have probably, I think I saw one of your guys' talks, someone had been talking about how when the viruses are more temperate, they produce more, like especially in bacteria is where we know it, but they produce virulence factors. So they're big animal, honestly. Did that work at all? Do you see where, so like in that big switch, that's what's going on. And then, everybody gets a little upset about like the increasing temperature, right? And so the increasing temperature is a problem because you cause microbes respond to increasing temperature just by increasing activity, right? Which also increases this sort of stuff. But we're also seeing a lowering of the oxygen in the atmosphere. So it's not just that we're increasing temperature, we're also lowering the oxygen in the atmosphere. We also have other things happening within these cascades right now, which are a little worrisome, let's say, from that point of view. And from a very Gaian point of view, like you don't have to believe that Gaia is living, but you definitely have to believe that there are feedback systems like Lovelock was talking about. That stuff is completely correct, right? You bury a whole bunch of CO2 as oil, and if you burn it, right, you're just going to go back to, this is how much, we had a whole bunch of extra oxygen and not as much CO2, and now we're going the other direction, right? We're trying to bring them back. Did that make sense, guys? Do you have questions? I know that's complicated, but it's cool.
Does anyone else want to jump in?
Well, just to say thank you for taking the time to explain these processes to non-scientists. I'm really grateful. Yeah.
It is actually like, you can see all this happening at whatever level you want to look. So I'll teach you something, like as a not practicing biologist. So one thing you can always do is, so I call it the Goldilocks line. So if you look at a system and you decide, do I have extra oxygen or extra sugar? That will tell you a ton about what's happening in the system. So for example, in your own body, you're a little oxygen low compared to the atmosphere, right? So you're spending all of your time. And if you want to build body mass, right, what you do is you go to the gym and you exercise and you cause the oxygen to go even lower. And so that's a biomass building thing. So anabolic and anaerobic, even though they don't mean the same thing, they actually are going the same direction. And that's how, and under those conditions, viruses tend to hang out with cells, right? But if you get immunosuppressed, right, and things start to go bad, the oxygen levels tend to go up, and that's because you've got like all this activity of neutrophils and so forth. So they're releasing oxygen-like molecules, and the viruses become active, right? So and you guys have all experienced it, like go in the sun, right? And get a whole bunch of sun, and you'll get like pimples or you'll get herpes outbreaks, right? And that's because of the reactive oxygen species that are around. So the microbes have switched, or the viruses have switched to lytic behavior. And that's like just kind of a universal thing. And so you can do that in any system, and you guys kind of know it, like everybody knows it. If you see clean water, like it's basically clear and it'll be relatively well oxygenated, compared to if you see like dirty water, it will be very low oxygen and it will be not something that you want to drink or get into, right? So it's just like basically you have it the whole time. So the viruses living in your colon are always temperate, with very few lytic events or virulent events. When the feces is released, it gets oxygenated and they become active, and they become virulent again. So that's just going on the whole time. So just always be looking, basically, how much oxygen do I have to how much sugar I have.
Thank you. So I've got one more question, just super broad, but if anyone else wants to have the last word, feel free to jump in and ask your question instead. So my super broad question is—
Go ahead.
Just, okay, I'm just curious what, just reflecting on popular culture during the pandemic, you've spent the better part of your life, thinking about viruses. And now everybody's got viruses on the brain. And you know, how, first of all, you know, do you see any receptivity to ideas that phage and viruses might be something other than pathogens? Like, do you see, like we saw early on in the pandemic, like sourdough bread baking, there was like, that was a thing. And so do you see like sort of the more probiotic idea of viruses gaining any traction in the pandemic? Or is this just like complete virusophobia? No. Short answer, no.
I'm just like everybody, like everybody's over viruses for right now. Nobody wants to think about the potential good side. No, honestly, you know, that and which is not, you know, not bad, right? That honestly people recognizing, like really recognizing this is the first time that that the planet together has really experienced what it's like, right? You know what I mean? Just because our communication is so good. Like HIV was always kind of a little hidden, even though of course it's been a little, you know, the most disastrous pandemic as far as we know. Like that's really, it was spotty, right? It was spread, or it is spread over like, you know, what, 40 years now, going on 50. But this one was so acute, right? Everybody saw it and everybody got a good feel for it. And everybody kind of knew that influenza could do this to us, right? But I do think that this has, like most of the world is actually responding reasonably aggressive towards it. And that's a good thing. Because this will happen, you know, it just will continue to happen. There's no, like, I mean, it doesn't really matter what your belief system is or anything, right? These viruses will just keep popping out and they're going to keep doing it. Like we're not, you know, well, I'm not that old. I'm kind of that old, but so I've been through 3 pandemics so far, right? You know what I mean? So it's not just like, it's like, you know, everybody, like I think previously people that even thought about viruses were, you know, kind of like, oh, the 1918 influenza was so bad. So hopefully this will spur research, and then people will see that the viruses that attack us can probably be countered by viruses that protect us, right? Because every virus that doesn't attack, there's a virus that doesn't want to die because of that attack. So I think that that it gives us a new direction to go with research, and maybe people will be supportive of it, or maybe we'll just say we don't care about viruses because they're annoying.
So on that note, we do have I'm going to butcher his name here, Konstantin Chumakov, talking about that in a few weeks, NIH researcher who's talking about the broad benefits of getting a live attenuated polio vaccine. You know, early on in the pandemic, he published a paper suggesting that we give everybody the polio vaccine to protect against SARS-CoV-2 because it basically has shown that you know, you get this live attenuated virus, and there's all kinds of different health benefits. So very soon we might be inoculating ourselves with good viruses, and yeah, this will start to, you know, transition from basic research science into, you know, probiotic cultures that many of us have already started to study in the realm of bacteria. So thank you, Forrest. This has been enlightening. I hope to continue the conversation. In the post-pandemic world, when travel is possible, I'd love to visit. And yeah, come back anytime you'd like to join the group. It's been a real delight to engage with this, and I love the book. The book is amazing. So thank you for creating that, and the artwork is also just fabulous. So many thanks, and hope to see you again in a Zoom room soon.
Okay. Thank you, guys.
Thank you.
Great. Have a good evening or day.
Yeah, all of the above. Thanks. Bye.
Goodbye. Bye.